¿Qué micrófono va a usar él?
Well, thanks everyone. It's my pleasure today to introduce today's speaker, Justin Touchon. And a little bit of a spoiler alert, I'm also briefly introducing Myra Huey, who's in the audience hidden today, but she's also going to be giving a seminar on June 6th. And they're both with us at EBD for a sabbatical stay. So I'm letting you know about them both because they still have like a little bit over a month left at EBD. So in case you're interested in interacting with them, talking to them, so they'll both be here for a bit more. So Justin and Myra, I've known them for when I was doing a postdoc at Boston University a long time ago. And we've collaborated over years in several systems. Justin and myself worked on local species in Massachusetts, in the States, in hatching responses to water molds and things like that. And after that, Justin decided to work mostly on beautiful tree frogs and leave those bufonids behind. Justin graduated, got his PhD from Boston University, and then did postdocs in STRI, in the Smithsonian Tropical Research Institute. Then briefly in East Carolina University, and then became a professor at Vassar College in New York, where she's a staff also at Vassar College. So he'll be telling us today about plasticity and the evolution of reproductive modes. He's also written a book on applied statistics with R that is actually a great manual for a lot of us working with R in ecology. And actually he's using part of this sabbatical stay to come up with the second edition of the book. So we'll keep an eye on that.
Thank you, Ivan. Yeah, thanks everybody for being here. It has been a lot of fun to be here for the past couple of months. And yeah, like Ivan said, we're here until the middle of July, so we have a little bit more time. And just to kind of go over a little bit again what Ivan said, we actually met about 20 years ago. So we were such babies back then. We were so young, and we did a lot of work in north of Boston studying wood frogs. And I was a brand new graduate student. Ivan already had his PhD. He was a postdoc, and he was a really amazing mentor to me. So over the course of many years, we worked on this system. We actually published 3 papers together. Ivan helped really coach me through writing my first paper as a grad student. He taught me so much about experimental design and statistical analysis. And and over the course of those years, we became really good friends. Right, such good friends that actually, when Myra and I got married, we actually came to Spain for our honeymoon, and we made Ivan drive us all over for like 3 weeks with his 1-year-old daughter in tow. But the most important thing, of course, is that everywhere we went, we found herps. So we had a really amazing time, and so we've been trying to figure out how to come to Spain for a sabbatical for a long time. So it's been really exciting that we were able to make it happen. And so what I want to talk about today is this idea, the role of behavioral flexibility and how that can influence evolutionary transitions, and specifically the evolutionary transition to reproducing on land. And as Ivan said, I've mostly left the boring, ugly bufonids behind, and I like to study these beautiful tropical tree frogs. So I'm going to try to take you guys into the world of this genus of frogs, Dendropsophis. This is about 100 species of small yellow and brown tree frogs found throughout Central and South America. And before we all of these guys, let's just start with one species. This is the species that I studied for my PhD. This is Dendropsophis ebricatus, known as the hourglass tree frog. They are a common lowland species. They're found throughout Central America, so you can find them as far north as Mexico and all the way down into Colombia and Ecuador. Like many frogs, most frogs maybe, I guess we could say, they come out at night. Males come to ponds where they're going to advertise to attract females. Females are going to come. They are looking for a mate. Here you can see a female, and she's trying to choose between these 2 males that are trying to court her. The mate choice in this species seems to be all really up to the females. They choose males. Once they have located a male they like, that male is going to hop on. He's like a little backpack. And she is going to hop around the pond to look for a place to lay their eggs. And what she's going to do is she's going to put those eggs up on a leaf above the pond, where they're going to develop really quickly. They hatch in only about 3 or 3.5 days. And then they're going to fall into the water below, where they're going to spend the next 2 months as a tadpole before they metamorphose and go back onto land. And so this life cycle, right, from adults that lay eggs, eggs that hatch into tadpoles, and then tadpoles that develop back into frogs, is really remarkable. And so before I get into the main topic that I want to talk about today, I want to tell you a little bit about the kind of other aspects of the biology of these frogs, because they really have plasticity. They have developmental, behavioral, morphological plasticity throughout all of their life. And so just to kind of touch on a little bit about these guys, we're going to talk mostly today about reproduction, but I want to start by telling you a little bit about hatching and development. So when we think about the embryos of this species, they actually have flexible hatching timing. So what you're seeing here is the time at hatching after oviposition for clutches in the field that were either protected from predators or were allowed to be attacked by ants and wasps. And so on the left side, we see eggs that were allowed to be attacked hatched about a third early compared to eggs that were protected. So they can hatch early to escape the threat on the leaf and fall into the water. Now there's a cost to doing that. If they hatch early, they hatch smaller, so they're less developed when they get into the water, but at least you survived that first risk, right? So they have hatching plasticity as eggs. Now, what I've actually spent the last 10 years or so focused on are the tadpoles of this species. Because back when I was a PhD student, one of the things I discovered is that if you raise these tadpoles in a nice, clean, happy, predator-free environment, maybe they grow up looking like this. But if you raise them with chemical cues of predators, so you have a caged predator in their environment, the smell of that predator, it's eating tadpoles, it's digesting them, it's excreting stuff into the water, that is going to cause that tadpole to grow a much deeper tail that has all this black and red pigmentation. If you raise them with a very different kind of a predator, a fish, so a dragonfly nymph likes to hang out in one place, it waits until something swims in front of it and then it lunges at it. If you raise it with a fish, which is a kind of a more actively hunting, chasing predator, they get this very clear, almost completely pigmentless tail that's long and slender. And so one of the things that we've been working on, we've known this aspect of how it changes their morphology and their coloration for a while now. We've more recently been studying things like behavior. So what this is showing you are tadpoles that were all raised as controls. They've never smelled a predator in the first 2 weeks of their life. And then we put them into an assay that either smells like dragonflies, smells like nothing, or smells like fish. And what we see is that after only 5 minutes of smelling that, if the water smells like fish, they start swimming more. If the water smells like dragonflies, they start swimming less. And so we have this very immediate effect on swimming behavior. Now, this is showing you that same assay, but for tadpoles that were raised for 2 weeks with fish or raised for 2 weeks with dragonflies. And so we see the same basic pattern. Tadpoles that were raised with fish just inherently swim more than those that are raised with controls. Tadpoles that are raised with dragonflies inherently swim less. And so then if we look at all combinations of development and then this, the behavioral assay, we see the same basic pattern. So tadpoles that are raised with fish, shown in blue, always swim more, no matter what water this. So we see kind of two time-scales of effects on their behavior. There's a developmental effect of the environment you're raised in, as well as an effect of the immediate environment that you're in. Now we also know that these tadpole phenotypes matter for survival. So this is showing survival in a 24-hour predation trial with tadpoles that were raised with fish and then were exposed to fish, or tadpoles that were raised with dragonflies and then exposed to dragonflies. So this is what you might think of as like tadpoles that have made a phenotype that is appropriate to a particular predator. And the first thing you can see here is that dragonflies don't eat as much as fish. Fish are a more voracious predator. They eat a lot more. But what happens when we put them with the wrong predator? So here we have tadpoles that are raised with fish, but then exposed to a dragonfly. And what we see is that their survival goes down. It's not that big of an effect because, again, dragonflies don't eat that much. But having the wrong phenotype hurts your survival. And we see with fish predators having the wrong phenotype, so being raised with a dragonfly cuts survival by about half. So having the right phenotype for a given predator improves survival. Having the wrong phenotype then conversely reduces survival. Okay, so that's just a little bit kind of to touch on like some of the other aspects of the biology of these frogs, which I think are really interesting. But that's actually not what I want to talk about today. Today we're going to focus on reproduction. And so when we think about reproduction, this is all based on stuff that I first came across when I was a grad student, and then I explored as a postdoc. And so when we think about reproducing on land, if we look across the animal kingdom, we really see that there are 3 phyla where animals have shifted from originally, evolutionarily laying eggs in water to then doing so on land. We see this in mollusks, in arthropods, and chordates, right? Animals basically began in the sea, and then we have an evolutionary shift to living and breeding on land. But when we think about the difference between these aquatic environments and these terrestrial environments, when we think about eggs, right, they're very different habitats. So how does this transition actually occur? How does an egg that is adapted and has evolved for surviving in water then survive in air, right? Because air is going to dry you out. There's less support, physical support around the egg. We think about how does a transition like this even occur? When we just focus on vertebrates for a moment, this is a very simple little cladogram, but we're all familiar, whether you study plants or not. I'm sure you know that fish breed in water for the most part. We think of amphibians generally doing the same. And then we have this shift to mammals, birds, and reptiles where breeding occurs on land. Now, some of you in the room certainly know this, but maybe many of you don't. Amphibian reproduction is actually much more diverse than you might originally think, right? We kind of think of frogs and salamanders as things being tied to the water, but actually different types of terrestrial reproduction have evolved literally dozens and dozens and dozens of times across amphibians and in frogs in particular. So a recent paper by Chris and Yvonne and others that came out in 2022 has this lovely figure which shows, if we look at the parts of these that are in green and brown, these are different forms of terrestrial reproduction. So actually different forms of laying eggs on land actually outnumber the number of species that lay eggs in water. So actually terrestrial reproduction is more common than aquatic reproduction when we look across amphibians. And that is actually going to be the case in the groups of frogs that I'm talking about. I showed you this earlier. I said that these animals can lay eggs up on leaves above ponds. What I didn't tell you was that this species, Dendropsophis ebricatus, can also lay eggs in water. Right, and so if you look at this and you're thinking that it doesn't look like they're in water, I'll point out that there's a tadpole swimming next to the eggs there. They're actually on a submerged leaf, laying their eggs directly into the pond. And so this is I just came across this in the field one day when I was a grad student, and we immediately started thinking like, okay, so if they can do both of these things, what causes them to do one or the other, right? What causes these frogs to lay eggs on land or in water? And so to test that, I learned how to sew, and I built these cages. I went into Panama City. I was working in the field in Panama. You buy these little children's swimming pools so you can make a little artificial pond. And then in that little artificial pond, I put 2 types of plants. I put plants that stick up out of the water and then plants that float. So then that gives frogs the option of laying eggs up on leaves above the pond or attached to the floating vegetation directly in the water. And then I placed half of these enclosures in the forest where there's lots of shade above them, and then half just out into and then in the morning you come back and see where did they lay their eggs. And so this figure shows the results of those first oviposition site choice trials I did. The green represents the percentage of clutches that were laid up on leaves out of the water. Blue is the percentage of clutches that were laid either fully in the water or at least in contact with the water. And so what we see is that in the forested sites, frogs are laying predominantly terrestrial eggs. Whereas when they're in the exact same type of enclosure, but it's out in an open field, they're laying their eggs mostly down in the water. Now the hypothesis here is that if you think about development from the perspective of eggs up on leaves, eggs that are going to be in the forest are going to be protected from the sun during that next 2, 3, 4 days while they're developing. Whereas eggs that might be up on a leaf out in an open site are likely just to get cooked. right? They're going to get dried out. They're going to be exposed to the sun. And so putting your eggs in the water allows you to protect them from the risk of desiccation. I'll point out one other thing, which is all of these decisions happen very quickly, right? It's in the course of a night. So these frogs are found in all these different ponds around where I was working. You know, males have been calling for hours. The female comes to the pond, they pair up, and then I come in and like grab them, drive them across town, and put them into one of these enclosures. And they still make these decisions in the course of, you know, a couple of hours. This is all occurring. So based on this, we call this reproductive mode plasticity, this flexible ability to actively change where they're, or even more broadly, just flexible behavior. Can flexible reproduction help explain the evolution of terrestriality or terrestrial reproduction? So, like lots of folks, I started with this one species, Dendropsophis ebricatus, and then I started wondering and thinking about close relatives that are in their little clade, and then I started thinking about, well, what about the entire genus? So this tree shows about half of all the different species of Dendropsophis. There's about 100 species or so. This tree has 54 species that have sequences available in GenBank. Species now from really across the tree. And so I'm going to recolor this. Let's add on what is published about aquatic versus terrestrial reproduction. And so what we can see, even just looking at this, so this is all just based on the literature, and hopefully if there's talk today, it's that you can't always trust what's in the literature. But this is based on what's in the literature, and what you can see here is actually there appear to be multiple origins of terrestrial reproduction within this single clade, right? Within this group of 100 species, it would appear that there's at least 4 independent shifts from aquatic to terrestrial egg laying. So the work that I'm going to describe for the rest of today, I was a postdoc in Panama at the Smithsonian Tropical Research Institute, like Yvonne said earlier, and I got to go to all these different places, from Mexico down to Madre de Dios in Peru. The 2 different colors here, orange versus red, indicate locations where I went to either work just with multiple populations of Dendropsophis ebricatus to try to compare within a single species versus sites where I could try to get as many different species of Dendropsophis as possible. This work obviously entailed a lot of help in the field. So everywhere I went, I got to have local assistance. And then also like some folks helped me that weren't even that local. So some of you might know Hyunji Lee. She actually did her PhD here at EBD with Yvonne and his research group. She was an undergrad in South Korea at the time and somehow found me and paid her own way to come to Panama and work with me, which is pretty crazy. And then Rosa Arribas also, while she was doing her PhD, got to come to Panama for a summer and helped me in 2 different sites. And so, like I said, so I worked in 8 different countries doing all this work. And what we did in each of these places was build a set of enclosures, a slightly modified design from the first one. This was a little more portable. So this was a 1 meter diameter pool, a 2 meter tall cage, and then in each site, wherever I was, whether it's Peru or Mexico or wherever, you find and collect locally, grasses and plants and things that will stick up out of the water, as well as floating vegetation. And then instead of trying to find forested sites and open sites in every one of these places, because each place is different, I would actually cover half of the enclosures in an 80% being in the shade. So all the cages are actually out in open areas, but then half of them are shrouded. I was in each site for between 2 and 3 weeks, and then it's the same idea. You go out at night, try to collect as many pairs of frogs as you can, you put them into an enclosure overnight, and then you come back in the morning and see where did they lay their eggs. Did they put them up out of the water? Did they put them directly in the water? Or like this, you can actually see the water line right here. This is a clutch of eggs that is essentially half out of the water and half completely submerged. So now we're starting to work, so in thinking about multiple species, the clutches of different species, the egg masses of different species can be very, very different, right? So this shows 3 different species, Dendropsophis serioquensis, triangulum, and ebricatus, and they have quite different egg masses, right? So this species, serioquensis, has a very globby, like thick, jellied egg mass that they attach to tree trunks and branches and things. Dendropsophis ebricatus just lays a very kind of thin sheet of eggs, a single layer on a leaf. Triangulum's eggs are kind of similar, but there's a lot more jelly around them. So I wanted to try to understand some of the more mechanistic differences in these clutches and kind of the physiology of the clutches. Larger eggs in general are associated with terrestrially breeding frogs. So if you look across all amphibians in general, species that lay eggs on land generally have larger eggs. It's because there's more oxygen available. A big egg has a hard time getting enough oxygen and water. But once we're on land, once we're in air, eggs generally seem to evolve to be larger. So larger eggs should be associated with terrestrial breeding, and thicker jelly should also help those terrestrial eggs prevent desiccation. So I did two things in each of these sites. So I quantified variation in egg size, and then I also tried to quantify the hydration rate of the jelly. So this is all done like in these like super remote places. So my methods might not be exactly what you would do in the most perfect scenario, but they're what I had at the time. So to measure egg diameter, I would remove as carefully as I could 5 eggs per clutch, and then photograph them with a digital camera, and then using ImageJ, measure the diameter of them in 2 different axes, which you then could average 5 eggs for a single female, and that gives you a measure of that female's egg size. And then to measure hydration rates, so I would take a cluster of 10 eggs with the jelly intact, and I would transfer it to plastic because plastic doesn't absorb water. And then submerge those eggs, and then periodically pull them out of the water, blot them dry, and then weigh them, which would give a measure of essentially how much mass the jelly is absorbing over time. So obviously this could only be done with eggs that were laid out of the water, because by the time I come back to my cage at 7 o'clock in the morning, eggs in the water have already been absorbing. So this is only done with species that laid enough eggs out of the water. But it should provide an estimate of sort of the maximum hydration rate of how quickly that jelly can take in water and hold on to it to keep those eggs protected. Okay, so I'm going to present sort of 2 different scales of analysis here. We're going to be looking, and on one scale we'll be looking across species of Dendropsophis, and then on another scale we'll be looking across populations of just one species of Dendropsophis ebricatus. But in both of these scales, I'm going to be asking essentially the same question, which is what is the role of flexible reproductive behavior on the evolution of terrestrial egg laying? And I guess I'll sort of take one moment to make sure my terminology is really clear, because so I've been throwing out flexibility and plasticity of an individual or a population or species to lay eggs both in water and on land. That is slightly different than plasticity, which is the active change in allocation from, like, let's say, a shaded to an unshaded habitat, right? So the plasticity implies responding to the environment and changing your behavior. So any species that has plastic reproduction is inherently flexible, but you can have species that have flexible reproduction that are not plastic, if that makes sense, right? So you could imagine a species that lays 50% of its eggs in the water and 50% of its eggs out of the water, but it doesn't ever change that behavior across environments. That would be flexibility, but without plasticity. Okay, so we go back to our phylogeny here. Like I said, I've actually done these experiments with all 13 of these species. So we'll start with this species up here at the top, Dendropsophis timbaba. So these guys, I was able to come across 6 pairs of these frogs down in Madre de Dios, Peru. And again, this is a species that is supposed to lay eggs only in the water, and yet in 2 of the 3 trials that I did with them in a shaded environment, they actually laid some percentage, about 1/3 of their eggs out of the water. Okay, so this is not a significant difference. We can't say they have significant reproductive mode plasticity, but it's clearly an indication that in some scenarios, under some circumstances, they can lay eggs, some of their clutch, out of the water, which is not something that anyone had ever seen before. So we can go back to our phylogeny, and now I'm going to add another color here. We're going to recolor this orange to indicate lineages that appear to have flexible reproduction. There was one more species that was very similar to this, Dendropsophis minudis. Again, I studied 8 pairs of these frogs, and in some of those trials in the shade, they laid some of their eggs, about 1/4, up out of the water. species that only ever laid aquatic eggs, never put eggs out of the water, so they actually did what the literature says they would do. There were 3 species that only ever laid terrestrial eggs. I will point out, if you look at these box and whisker graphs here, you can notice there's not very much there. These 2 species, Dendropsophis bokermani and Dendropsophis frosti, are very, very rare. I only came across 1 pair of each of them. This species, Dendropsophis frosti, was actually, I think I'm only the 2nd person who had ever seen them in the wild. They were actually the species was being described when I came across them. So if anybody knows Santiago Castroviejo Fisher, I know he's a Spaniard. He was the first person to really discover them and was describing them when I came across them, and also got to at least throw 1 pair into my enclosure. But we're going to say they seem to have terrestrial eggs as far as we can tell. Now there were another 4 species that all had varying degrees of flexible reproduction, including 2, Dendropsophis ebricatus and then also Dendropsophis bifurcus, that actually did have a significant change, right? They lay significantly more eggs in the water in unshaded environments, or vice versa, more eggs out of the water in shaded enclosures compared to anybody else. Now, that's a whole bunch of box and whisker graphs, so to kind of pull that all together, I'm going to plot all those in terms from thinking about from the most aquatic species to the most terrestrial species. And what this really shows is that there is a continuum of degrees of flexible reproduction in this lineage, from species that are mostly aquatic but will sometimes play with their eggs in the water under certain circumstances. All right, so I'm going to redraw that phylogeny just with the 13 species that I've actually studied, including, and then an outgroup. Here I'm going to show estimates of ancestral state estimation. So we can recolor this based on the probability of the percentage of eggs laid either in or out of water in evolutionary history. And so what we see here is, from an aquatic origin of the whole lineage, we see different groups where we clearly have an indication of flexibility that leads to extant terrestrially breeding species. So this would imply that plasticity, or flexibility rather, can provide the bridge between only laying eggs in the water and only laying eggs on land. These pies indicate the probability of one of the sort of 3 states at any node in evolutionary path, so essentially having aquatic reproduction, terrestrial, or having flexibility. And so again, what we see is that there are nodes where it's most likely that there's aquatic reproduction in the past, but then we have lineages where it's most likely to have flexible reproduction on the way to extant species that lay eggs only on land. Now, that's all about the behavior. What about egg diameter and the jelly hydration rate, as I was talking about? How does that vary across species? Well, what we might expect when we think about egg size is that we would expect that more terrestrially breeding species should have larger eggs, right? If we see this general pattern across frogs in general. And that is actually exactly what we see. So the little dots here are individual females, and then the larger, we see this large macroevolutionary pattern that's common across amphibians in general, also plays out in this group and really tracks flexibility in reproduction. Now, when we also, that's us. When we think about the hydration rate of the jelly, we might expect species that are more terrestrial to have jelly that absorbs water more quickly and also can hold on to more water. And again, that's what we see. So the one terrestrially breeding species here is Dendropsophis serioquensis, and their egg jelly can not only absorb water more quickly than these 3 species with flexible reproduction, it can also hold significantly more water. It just can physically retain more water in the jelly than these species with flexibility. So that's going to be an indication of adaptation to life on land for those eggs. So when we look across these species, the macroevolutionary pattern seems very clear, right? We have essentially flexible reproduction as a bridge between only laying eggs in water and only laying eggs on land. And then as those species become more terrestrial, we see the evolution of these other adaptations for life on land, right? Larger eggs and an egg jelly that is essentially enhanced, that is better for protecting embryos from desiccation. Now, what happens when we look across populations of one species, across Dendropsophis zebrocatus? And this map is showing you, the colors here show rainfall. So we can see that up here in Mexico, it is a bit drier than farther south, particularly here in the Choco of Colombia, which unfortunately I did not get to go work in. What?
No.
Yeah. So anyway, we might expect a pattern across that rainfall gradient that would also affect the degree of terrestriality and reproduction. So pretty much every population has flexible reproduction. There were 2 that actually had, again, what we would call plasticity, that are actively switching to a significant manner, whatever we want to call statistical significance. But there are 2 populations in Panama, Rambala and Gamboa, that significantly lay more eggs out of water in the shaded conditions compared to unshaded. But the pattern is the same, even in pretty much all the populations that are not significantly different. We see more eggs in general being laid out of the water in shaded conditions. Now, what's really amazing is these 2 populations, in Piro, which is in the Osa Peninsula of Ecuador, and then down in Durango, Osa Peninsula of Costa Rica, and then Durango, Ecuador. We have 2 populations that have essentially evolved terrestrial reproduction. They basically never lay eggs in the water, or very, very, very few. So that's really interesting. So we have 2 populations out of these 8 that have essentially seemed to have gone almost all the way to fully terrestrial egg laying. Now, these are all single species, so instead of trying to put them in the same kind of phylogenetic context, we're going to look at more of a population genetic scale. So this is a haplotype network of those 8 populations, and I'm going to walk through it, but then I'll point out the one really important takeaway. So here we have, these are our 2 populations from Panama. This is sort of the far north, Belize and Mexico. Here are our 2 populations from the Caribbean coast, Costa Rica and Nicaragua. But the real takeaway here is this haplotype network is based on 1,400 base pairs of mitochondrial DNA. And this Ecuadorian population, 213, that represents the number of base changes from Ecuador to Central America. So about 1/7 of that chunk of mitochondrial DNA differs between Ecuador and the rest of Central America. So that population in Ecuador is very evolutionarily divergent from the rest of the species range. Now, with 6 of these populations in Central America, just because of permitting reasons, I was able to get more tissue for those. So we actually also did rad-seq. So this is a structure plot based on about 52,000 per population. It's 10 individuals per population. And what this really reveals is that we have a cluster. So the 2 Panamanian populations are clearly a single evolutionary lineage. Our 2 populations in the north, in Mexico and Belize, also are a single population cluster. But the 2 populations in Costa Rica, the Caribbean and the Pacific side, cluster are unique individual lineages. So that population in the Osa Peninsula, Piro, that is breeding terrestrially, is an independent lineage from the rest of this species. Now, when we look at things like egg diameter, we see there's actually no relationship between the size of the eggs across populations and the degree of terrestrial reproduction that we have. So there's no relationship between egg size and degree of terrestriality. And when we look at the jelly, though, we see that one population does seem to have enhanced egg jelly. So the population in Ecuador has jelly that is absorbing water more quickly and can hold on to more water than that seen in the rest of Central America. So, what else can explain variation in the reproduction across these species? Well, when we just look across them in general, latitude is a very strong predictor of terrestrial reproduction. So populations that are closer to the equator lay more terrestrial eggs. Populations farther from the equator lay more aquatic eggs. Part of that is driven by variation in rainfall, which is shown here. So this is rainfall during the warmest quarter of the year. And so again, populations that are drier have more aquatic reproduction. more terrestrial reproduction. And then lastly, more thermal stability. So this is a metric from the World Clim 2 database called isothermality, which is the ratio between DL temperature and annual fluctuation. So it's basically a measure of like how stable the climate is. And again, so our most stable population, Durango, is also where we see the most terrestrial reproduction. Okay, so that's a lot. The story with Ebracadus is very kind of complicated, so let's walk back through it. First, we have two populations that have essentially evolved terrestrial reproduction, right? They've basically stopped laying eggs in the water. Those two populations seem to be evolutionarily distinct from the rest of Central America. They are not sharing, there's no gene flow between Ecuador or between Piro and these other populations that we've studied, or that I've studied. One of those populations, Durango, Ecuador, also has evolved an enhanced egg jelly, but none of those populations have actually evolved differences in egg diameter yet, in egg size, this thing that we see across species. And those populations that have evolved to be more terrestrial are closer to the equator and are in very thermally stable and wet regions. Species to transition from only laying eggs in the water, from aquatic obligate reproduction to laying eggs on land. That there's this period of flexibility when species can do both that allows them to really get all the way out of the water. When we look across these species, we see that species that have terrestrial reproduction also have larger eggs and have the jelly that surrounds those eggs is a terrestrial egg laying, but only one of those populations has evolved any differences in the egg jelly, and no populations have evolved differences in egg size. So what that really tells us, the most important thing here, that tells us that behavior is actually evolving first. Behavior is leading the way in this major evolutionary shift from water to land. And then these more kind of physiological and morphological adaptations come secondarily. So they basically catch up once the behavior has really kind of paved the way. And so with that, I think it's 1:45. So I'm just going to say thank you to all of you for listening. Thank you to the NSF for funding all that work. My advisors at STRI, Harris, Roberto, and Owen. And thanks to everybody that helped out with all of this work across all these countries. And yeah, I'd be happy to take questions.
Inspiring. Thanks very much, Justin. Looking forward to some questions from the audience. I have many. But, I mean, the question that I always have, and I'm sure you've thought about in your system, is why are they laying eggs out of water in the first place?
Mm.
Like, so shaded versus open seems to allow them, or one condition is more favorable than the other.
Yeah.
But why start laying eggs out of the water in the first place?
Yeah, so data that I didn't show, which is also stuff that I did as a postdoc, shows that, so if you put frogs in the open, but then you put fish or other kinds of predators in the water, that actually will cause them to lay eggs out of the water. So from the frog's perspective, clearly the risk of predation to their embryos is greater than the risk of desiccation. And so that would be my answer, so is predation risk. You're trying to, because if it rains, your eggs are in good shape. And yeah, there's predators on land, but there's fewer than if they're in a pond, like the predation rate is, and I also have data, I have stuff that shows that the predation rate in water is much higher than on land. Yeah.
Thank you very much. Is very, very, very deep in knowledge of this group of frogs. I have several questions, but one should be more general. For instance, I was thinking that other groups of frogs, like leptodactylics, they also can breed in land or in water. These form nests, especially Leptodactylus, Angistomus, and so on. But obviously, they have foam. They have an innovation, evolutionary innovation that probably allow more possibilities. But yes, what's your opinion about in this other group that you are not expert of course? me too. But yes, thinking is because you have in leptodactylid, you have species that make the fawn nest in a underground cavicola group, for instance, in northern Argentina, for instance, and others put in land, others put in water, others put in water, and suddenly the pond dry out, and so on. No? You have, but yes, they don't use obviously this possibility of the jelly absorbing. But I don't know if you can compare these two groups. Basically, it's a very light question, as you can imagine. But
yeah, but I think that the commonality there would be like this idea of behavioral flexibility. So the foam nest is really amazing for protecting them, right? It protects the eggs from drying out and does a really, really great job. But the idea, I mean, maybe I think the common thread between them would be, imagine a species that, we definitely think about them as, Tungara frogs or whoever. Some ability to flexibly decide between some of those options, then that creates other possibilities that could evolve secondarily. So I don't think of it as actually necessarily that different as far as that idea that having flexibility in decision-making or in the behavior can still lead the way between some of the differences you see amongst leptodactylids or other groups. Yeah.
Second question is more concrete. If, for instance, do you find or have you found a difference in developmental rate for those?
Any of that on these with this stuff. The data that I do have on that, so from my PhD, we did Ebercatus eggs developing in water versus on land, and yes, they develop more slowly because there's less oxygen, and so they develop more slowly, but then when they do hatch, they are actually still smaller, even though they've developed longer than the ones in air. Yeah, so I'm sure that it affects them. But then it's also complicated because so the species Dendrosophis stereoquensis, their eggs are just noticeably, just from the naked eye, you can see their eggs are larger than another one of those flexible species or the aquatic species. So then it's, it's just complicated by those other things, right? It's not just straight development time, I guess. Yeah.
Okay, thanks.
Yeah.
Thank you very much. You had your experiments were this like shade, no shade. In a smaller scale, taking into account the landscape, the very local variables, would you think that will stand, or do you think there will be more subtle differences in laying eggs on the...
Like what other things might cause them to switch behaviors?
Yes,
or?
In the local environment.
Yeah.
Higher temperature, lower humidity.
Certainly, like I said, I don't know what cues they're using to make decisions, right? So we can cover the thing with a cloth, and that seems to affect their behavior. But is it actually, is it light? Is it temperature? You could imagine in a forested site there might be less wind. So that should actually dry out the actual adults differently. Like if it's really windy in an open place, that should dry out the adults. So we don't know what cues they're using, but certainly there could be lots of things that affect those decisions, yeah. But we don't know what they are yet. Yeah.
Any more? Okay. Can you pass it?
Yeah, thank you very much. So, do you have any estimate about the heritability of flexible behavior?
Not for reproduction. So, I've done a study looking at heritability of the tadpole plasticity, so the ability to respond to dragonflies or fish. And it's very heritable, so the actual ability to be plastic is very heritable. But for reproduction, no. But I guess I could try. So I actually have a captive colony. I have a colony of Ebercatus in New York at Vassar College. And, but takes about a year for them to go from egg back to adulthood. So we could get those measures. Like we actually could try to get that over the course of many years. But we don't. I don't have an estimate of that right now. No. It's good. It'd be great to have, though.
Thank you. I was wondering if you, when you were collecting the frogs, did you notice a greater abundance of those that you know have flexible reproduction routes? Like, would you say having that flexibility is like more successful as like a species or population group?
Yeah, that's a great question. I mean, the thing that I will say about that is like, let's see here. Let me find this map. I mean, well, this will work. So this species, Ebercatus, right, that I studied, I mean, they are like, I have friends from Panama who would refer to them as trash frogs. Like you can find them in the grossest garbage ponds or cattle pastures or whatever. I mean, they are everywhere. And they have this tremendous flexibility, right? They can lay eggs above water, below water. They can breed anywhere. So does that make them more successful? I don't know, because there's other species that kind of co-occur with them that are only lay eggs in the water. But they're certainly very successful for what they are. Yeah, and there you just find them absolutely everywhere. So maybe, yeah.
Great, thanks.
Any more? Have one.
Yeah.
When you plot like comparison between species,
Mm-hmm.
you say like were the two more plastic ones, also like the more terrestrial ones within the flexible species?
Uh-huh.
Like I don't know whether it's just because you plot it there or like.
You mean this
so
one?
is it play? Yeah. So is plasticity a terrestrial trait that has been selected?
You mean like the idea that these two
Being
are
the
sort of
most
the most terrestrial of the flexible ones and they have plasticity? I think that if I had a larger sample size of these guys, we would have certainly detected plasticity. You know, the pattern is certainly the same, even if it's just a small sample size. These guys were essentially, in most of these species, this guy Timbeba, like I never thought I was going to find them. I was in that site to try to find this other species, Leucophalatus, which I only ever found 4 pairs of. And so it was just like whatever I could find, be like, oh my God, throw them in the cage, let's use them. So I think if I had a larger sample size, we certainly would have called that plasticity. So I think there's not necessarily a reason to think that plasticity would be associated with the more terrestrial kind of thing, yeah. Yeah.
Any more questions? Well, they are for 1 month more, so
Yeah.
any more questions, you can always contact them.
That's right.
Thanks again, Justin,
Thanks.
for presenting here, and see you all next week. There is since Thursday is holiday, we will have one extra speaker on Tuesday. Okay, so we will send an email anyway. See you next week.